Okay. Welcome to the biological life sciences students. Today, on the demand of one of my subscriber, I am making this video. That is the video on SDS page. That is sodium dodecyl sulfate polyacrylamide gel electrophoresis. Okay. So let's begin with the video now. So you can see here. Let me discuss the sodium dodecyl sulfate here. First of all, it is a type of electrophoresis. Remember, it is a type of electrophoresis which is always done to separate proteins as we separate the DNA on agarose gel. Okay, so agarose is a polymer. It is made up of agarose is a disaccharide which is made up of galactose and 3,6 anhydrogalactose. Okay, so similarly here also for the separation of proteins we use SDS page. SDS is an anionic detergent here. Remember, SDS is an anionic detergent that is having negative charge. Okay, and page here means polyacrylamide gel electrophoresis. Okay, so it is made up of acrylamide. What it is made up of? Acrylamide. Okay, so what is acrylamide? Acrylamide is a potent neurotoxin. Okay, remember, acrylamide is a neurotoxin. So we use, why we do we use acrylamide here? So acrylamide polymerizes better. It has stability. Therefore, we use two types of polymer here. One is acrylamide and second is bisacrylamide. Acrylamide and bisacrylamide. So the gel which is formed in SDS page is acrylamide and bisacrylamide gel. Okay, so here this is a kind of machine that is shown here. This is vertical gel electrophoresis machine. Remember, vertical, it is vertical gel electrophoresis. It is available in various shapes and sizes depending on the need. It is of different types. You can see here one SDS page gel is shown here where the protein bands, you can see different protein bands that are visualized here, and this is stained by Comasi Brilliant Blue. Okay, Comasi Brilliant Blue staining is done here. So let's see in details how this happens and what are the different properties of SDS page and what are we going to learn here. So we are going to learn the principles that is required for separation of protein by SDS page, then how one can prepare and run SDS page gel, and then how SDS page gel are analyzed. I will give you the composition of acrylamide and bisacrylamide required for preparation of stacking gel as well as separating gel. I'll give you a typical composition, but it may vary depending on the use case. The cases in which you are using it, different cases for separation of proteins. Okay, first of all, remember the protein that are separated on the SDS page gel, it should be in the primary form. Okay, it should be in primary form. Why it is necessary to be in primary form? Because in primary form the mobilization is proper. Okay, the mobilization of the proteins are proper. Okay, so you can see here, this is the protein in a quaternary or tertiary form. Okay, so it has to be converted into primary form. Okay, and it can be done only using heat, that is denaturation. Okay, now protein is having different types of charges. Okay, it can be negative, it can be positive, or it can be neutral also. Different kinds of proteins are there. Okay, so now when you have to separate it in electrophoresis, it should have a particular type of charge. Okay, so for that purpose we are adding the SDS here, that is an anionic detergent, or that is negatively charged detergent that imparts the negative charge to the proteins. You can see here it imparts the negative charge to the proteins so that the proteins will move to the positive side in the electrophoretic chamber. Okay, so therefore SDS, it helps in maintaining a constant charge in this kind of electrophoresis. Okay, so let's see the different cross-linking agents here. You can see the bisacrylamide here. Okay, this is the bisacrylamide uh, you know monosaccharide here, and this is the sorry monomer here, and this is the acrylamide monomer here. You can see the structure shown to you here, that is actually helping in this polymerization. Okay, so you are using two different acrylamides. One is N-N-methylene bisacrylamide. That structure is shown to you here like that. It is methylene because of this presence of methylene group. Okay, then you can see the acrylamide structure here. Okay, so now initially these two are separate. These two components are separate. Okay, in a solution and they are mixed together, they still remain separate. But when you add timid and ammonium persulfate, it starts catalyzing or it starts polymerizing. Okay, now here the ammonium persulfate, it provides the free radical that initiates a vinyl polymerization reaction. Actually, it is a vinyl polymerization reaction and it is initiated by the persulfate radical. It is initiated by persulfate radical. Okay, so remember this persulfate radical starts the polymerization of the gel. Okay, you can see here the mixture of the two are formed like this. Okay, so here the base acrylamide is generally used in cross linking. Okay, you can see here the cross link formed by the base acrylamide, and the acrylamide forms a horizontal linking like this, and this is the vertical linking that results in the formation of gel of this kind. Okay, it will form a polyacrylamide gel of this kind. Why polyacrylamide? Because the number of acrylamide molecule taking part in the gel formation is more the concentration of acrylamide that you are using, more will be the smaller will be the pore size. Okay, okay, the more amount of concentration of acrylamide, smaller will be the pore size, because the concentration will result in a more meshy network that will create a gel with a smaller pore size. Okay, so the concentration of acrylamide is generally adjusted depending on the molecule of proteins that you are going to separate. If the molecular size of the proteins is very large, then the pore size must be more, so the acrylamide concentration should be less. Okay, so the generally polymerization reaction takes place only after the addition of ammonium persulfate. Now this reaction is very slow. Okay, it will take more time if we don't add timid. Okay, so what does timid does here? Timid increases the polymerization rate. Timid increases the polymerization rate and helps the gel to polymerize faster. Okay. There's a caution here. The unreacted acrylamide and bisaccharamide monomers, these are neurotoxin. I already told you earlier. So therefore, it has to be dealt very carefully. Okay. So generally, native page is there, or the gel which is having only one kind of page, that is polyacrylamide gel, is native page. But when you are using a separate two different gels or discontinuous gel, it is having stacking and running gel with different compositions. Okay. So you can see here the stacking gel is kept at the top of the gel plate in the gel where you are forming it, and the this one resolving gel is placed below it. Okay, so there's the composition of these two are different. The stacking gel and the function is also different. Stacking gel and this resolving gel, or it is also called as separating gel. Okay, the composition is different, plus the pH difference is also there. This pH difference helps in separation of protein. Now I am going to tell you exactly how the proteins are separated based on this. Okay, the proteins are generally, it is loaded onto this well and it stacks together at this level. All the proteins comes together here when the electrophoresis is happening, and when it is separating, when it comes to the separating gel, it starts separating over here. Okay, generally here 5% and 12% acrylamide solution is shown to you here. I will give you the composition of one of the acrylamide. If you want, you can write it. Okay, so first of all, I am giving you the composition of the separating gel. Okay, So, sorry, say stacking gel, the upper one, the stacking gel. Okay. So let's begin with the composition. The first of all, you have to take double distilled water because you want it purified. Okay. I'll give you the composition for about 20 ml. Okay. You can reduce the concentration as per your convenience or how much you want, how much gel you want to prepare. Okay. And the 1 molar, there will be tris HCl. The pH of this tris HCl should be adjusted at 6.8, and you have to take this as 3.15 ml. Okay. Then 40% acrylamide solution is to be taken. Okay. Then this 40% acrylamide solution, you have to add 3.13 ml in the gel mixture which you are preparing. So you have to take a beaker and you have to assemble everything here in this beaker which I am telling you. Okay. One is double distilled water, second is tris HCl, third is 40% acrylamide, then fourth is 10% of SDS. It gives the positive charge. Okay. So SDS you have to use 0.25 ml. Just a minute. Okay. Then you have to use 10% SDS, then timid. Timid concentration should be 0.025 ml. Then 10% APS solution, ammonium persulfate solution. You have to use it as 0.25 ml. Okay. So this is the composition of stacking gel, the gel which you are going to pour at the top. Okay. Now let me give you the concentration of uh, separating gel. Okay, separating gel acrylamide concentration is generally more because you want to separate the proteins. Okay, the proteins are separated on the basis of charge and the proteins are also separated based on the molecular weight. Okay, so let's see the separating gel. Okay, so this is the I'm giving you the standard type of gel. You can vary this depending on the protein this protein that we are separating. Okay, remember I'm giving you as a whole 100 ml gel. Out of that we have seen 25 ml stacking gel and the 75 ml separating gel. Okay, so the separating gel here which I am giving you for is for 75 ml. The stacking gel which I gave you concentration is for 25 ml. Okay, so generally the stacking gel separating gel ratio, it should be around 1/4 to 3 by 4th. Means 1/4 is if it is stacking gel, then 3 by 4th it should be separating gel. Okay, sometimes it is also 2 by 3rd of separating gel and 1 by 3rd, that is stacking gel. It depends on the machinery, also the plates which you are using. Okay, so here double distilled water, it has to be 35.85 ml in 75 ml total, of course. Then it should be 1.5 molar tris HCl. Here the pH should be 8.8. It is 18.9 ml. Okay. Then 40% acrylamide mix. This has to be added at 18.75 ml. You have to note that the concentration of this is more here. Okay. Then 10% SDS. You have to add 0.75 ml. This is important as it gives the charge and it denatures the protein. Then timid. Timid you have to add 0.03 ml and then 10% APS 0.75 ml. Okay, remember the timid and APS has to be added at last. Okay, in the upper part also this has to be added at last. Okay, because once you add this, the polymerization will gradually start. Okay, so this is the composition of the both the gels as shown you here. Okay, and you can vary the concentration also depending on the sizes you want. Okay, the protein migration varies greatly in stacking and gel depending on the concentration. Okay, the running gel that you are using here, or the running buffer which you are using, electrophoretic buffer, is generally tris glycine SDS buffer. It consists of glycine. Okay, consists of glycine. Okay, now let's see. Here, generally in stacking gel, both amino and carboxy terminal are charged in stacking gel, that is pH 6.8. Amino group loses some positive charge in the running gel, that is separating gel, 8.8 pH. Okay, so here the at the pH 6.8, the amino acids is generally in this form. You can see the zwitterionic form, where both the charges are same, or the total charges are neutral. Okay, so glycine is used here because it is a very small amino acid, plus it helps in charging intention also. Tris glycine is yes buffer, pH 8.3. The protein samples are loaded here. Okay, you can see the protein samples are loaded here. The tracking dye, or the sample loading dye that is used in proteins, is generally having bromophenol blue. Then it is having uh, glycerol or sucrose. Yes, this gives the density. Okay. Okay, so this gives the density. Now let's see, when the voltage is applied, what happens? The chloride ions in the stacking gel buffer moves rapidly to the positive pole. Okay, sorry for the disturbance. The chloride ions in the stacking gel buffer moves rapidly to the positive pole. Okay, so the chloride ions which is already present here in this, it starts moving to the positive pole. Okay, you can see here. Okay, now the glycine molecule along with it enters into the stacking gel. Okay, it enters into the stacking gel from here. Okay, it is already present in the buffer which is placed in the electrophoretic tank. Okay, so the protein are negatively charged, runs at various rate through the stacking gel. The proteins are of different charges because negative charge, but depending on the molecular weight, it runs differently. But what happens? It starts stacking at this position. Okay, it starts stacking when it reaches here at the interface of the two gel. Okay, glycine moves slowly through the protein, slower than the protein. So glycine is lagging behind, and the protein is, suppose this is the glycine, it is lagging behind, and the protein molecule is stacking here. Okay, since very few glycine molecule have negative charge. Okay, now what happens when this all the proteins are at the interface? It starts to move. Okay, so the differential migration of chloride and glycine ions sets up the potential difference that helps to the concentrate proteins. The glycine ions lags behind in this gel, and the chloride ions is moving forward. Okay, faster. Okay, here glycine is there, it will be lagging behind in this area. Chloride ion will be in this area. Okay, faster, it will move faster. So there will be a potential difference that is created here in this region. Okay, due to this difference, what happens? The protein stacks up at the interface between the two gels. The protein will move definitely, but when it reaches at this position, it stacks up, it stacks together. Okay, now when it tends to enter into the separating gel, okay, migration of protein, when it tends to enter into the separating gel, the glycine amino acid loses protons as it enters the separating gel. This extra proton which is present, it starts losing it. Okay, so now negatively charged glycine which has lost the proton moves rapidly than the protein at the pH of running gel. Okay, the glycine moves faster now. The proteins now, when the potential difference is removed, all the proteins that are stacked at this position starts to separate depending on their molecular weight. Okay, it does not separate on the charge. Remember, the glycine moves faster, and due to this, the potential differences, or the potential difference that is created here at this position is disturbed, and the proteins starts moving according to their molecular size in the running gel below. In the running gel below, protein starts moving depending on the molecular size. Okay, so migration of protein that is invisible in the running gel is inversely proportional to the log of molecular weight. Okay, the molecular weights log, it is inversely proportional to that. Okay, so let's see how it happens. Okay, the chemical that we used, obviously I told you the composition also, the acrylamide solution, then stacking gel we have to prepare from it, then we have to prepare running gel, ammonium persulfate at timid, and we add these two things at last. Okay, so you can see the gel plate here. It is having a spacer, or the gel plate is having the you know an rising here. It is generally thicker at this position and thinner here in this position, which gives a 1 gel. Okay, so in some cases, in some units, you get a spacers also. Okay, Teflon spacers you get. Okay, to create that gap of 1 it is also available in 0.5 gap. Different companies which makes this unit provides you the different thickness of the gel. Generally it is 1 or 0.5 thickness gel. Okay, second plate is shorter because it is having the same width. So you can see the plate here, second plate, it is plain and it is having certain uh, elevations which will make a cavity of 1 here. Okay, these plates are generally very fragile because it is made up of glass, so, and it should be clean. Okay, so you can see this kind of assembly you can use, or you can use other assemblies also provided by different companies. Okay, so you have to first of all seal the all the areas, all the sides, to avoid the leakage of the gel. You have to seal all the sides. Okay, now casting is done. Casting of the gel is done. First of all, you have to pour the separating gel. Okay, you have to pour the separating gel first, then stacking gel. Okay, so the stacking gel will be at the top. Okay, you have to mark here, and then the stacking gel preparation. So while you pour the stacking gel, then you have to add the comb. Okay, so after every pouring, remember, just wash it with the deionized water. Okay, why you have to wash it with the deionized water? I'll tell you, because the process of polymerization, it happens quickly, plus oxygen is inhibitor of this process. Okay, oxygen is inhibitor of this process. Okay, so therefore you have to, oxygen inhibits polymerization, so therefore you have to avoid the air bubble, plus you have to put water in this layer when you have poured the gel. Okay, so over the gel will thin layer of deionized water, and you can remove this deionized water using tissue paper and all, and then you have to put the next gel and wait for polymerization. After the polymerization is done, you have to load the protein sample here. I told you that the protein sample has to be boiled for at least 10 minutes. Okay, since it makes it in a primary structure form, it will make it in a primary form. Okay, and that is important for electrophoresis, we have seen. Okay, so once you do it, or once you separate that, uh, then the samples is ready for loading. Okay, remember, avoid trapping the bubble at any cases. Okay, and when you put the comb like this, combs are available like this, it will make a cavity in the stacking gel, and you can load the protein sample here. This is the electrophoretic apparatus. You have to place the gel into that electrophoretic apparatus and remove the comb carefully. Now when you remove this comb, remember, I'll show you, when you remove this comb, you can see this is the cavity that is creating the wells here into which you are going to load the protein sample. Okay, now when you are removing this comb, what happens, a vacuum is created in this area, so it has to be removed carefully. Okay, to avoid the collapsing of the wells, you just add deionized water at this interface. You just add deionized water. Okay, so when you add the deionized water, it will take the area, or it will go into this, seep into the gaps, so that the wells are not destroyed, because air tends to go here and it may discard your wells. Okay, so this is the technique that you have to use when removing the comb slowly. Just add water, deionized water at this interface, and slowly remove the comb. Okay, after this, you have to add the reservoir buffer. I already told you the reservoir buffer is trace SDS glycine buffer. Okay, in many teaching kits, it is just named as reservoir buffer. Okay, but it is actually trace SDS glycine reservoir buffer. Okay, so when you pour this, then you have to load the sample. Now you can see the tip here. It is a different kind of tip. Okay, it is hairy root tip. Okay, it is very fine, and it is lodged into this well, and slowly the protein sample is added into that. Okay, and when the protein sample is loaded, the electrophoresis is started. Okay, when the protein sample is in wells, you have to put the electrophoresis at 50 watts, and when it crosses or it enters into the gel, you can move it to 100 watts, and you have to do the electrophoresis for 100 watts. Generally, the tracking dye that you are adding should be at the bottom of the separating gel. Then only the electrophoresis will be over. Okay, okay. So generally it takes about 1 to 1.5 hours. Okay, it depends on protein samples also, obviously. Put the power supply properly and then start the separation. You can see the separation here, and as I told you, yeah, when it reaches the bottom of the gel, that is tracking dye, you have to stop. Okay, so once this is run, the gel or the SDS page is over, you can see the gel looks like this. Okay, you have to carefully remove this gel and put it in a tank. Okay, it can be any container where you have to stain it. Okay, generally the proteins are here in this area, and you cannot see the proteins, so you have to stain it first. Okay, so the traditional, that is the dye that is used, is Comasi Brilliant Blue G250. This is the structure. You can see the structure of this dye. Okay, this is this dye. Gel are rinsed with water several times to remove the gel chemicals, and then they are incubated with simple blue, a colloidal solution of Comasi Brilliant Blue or Comasi Blue. Okay, so after you have stained it, you have to destain it by adding destaining solution, which generally contains acetic acid that removes the background stain. Okay, background stain is removed, and only the protein bands will be stained with the blue color, and you can see that. Okay, actually this is the black and white image, but the proteins looks okay, remember, blue in color. Okay, now these bands looks blue in color. I already shown at the top. At the top I have shown you. You can see. Okay, not this one. Wait. This. When stained with Comasi blue, the protein looks like this. Okay, the bands look like this. Okay, you can alternatively stain it using silver staining procedures, which is more sensitive. Okay, silver staining procedures is more sensitive. It generally has silver nitrate or silver chloride solution. Okay, okay. So silver staining is done. I'll talk about this in a different video, but silver staining is done for more sensitive protein molecules, where the amount of protein molecules is very small, or the band is very faint. So in such cases, silver staining is done, but for general purpose we use Comasi Brilliant Blue only. Okay, remember. Okay, the diagram that was the photo that was shown here at the last was actually of a silver stain. The photo that I have shown you, this one, this is actually of a silver stain. Okay, so this is all about the SDS page. I hope you have cleared every doubt that was there regarding the SDS page. If any doubt further, you can comment below. Okay, feel free to ask any comments, question in comments. Thank you for watching. Okay, do consider subscribing my channel for more such videos. Thank you.